Gene editing is a technique used to precisely and efficiently modify DNA within a cell. This enables addition, removal, or alteration of DNA with the result of changing its function. So, how does it work? First of all, a gene is a short section of DNA that codes for a particular protein, and the most common system used for editing these genes is CRISPR-Cas9. It uses a type of engineered nuclease. These are essentially molecular scissors, and these scissors cut the gene in a specific place. After the DNA has been cut open, repair enzymes will naturally try to repair the cut, but we can manipulate this repair process to make various edits to the DNA, such as insertions or removals in exactly the place we want.
But how will this revolutionise our lives? 1. Primarily, gene editing could make radical improvements to human health. If it fulfills all its promise, CRISPR-Cas9 could eradicate family lines of hereditary single-gene disorders, such as sickle cell anemia, a disease where people have abnormal hemoglobin, which in theory could be cured by a single base pair change in the DNA of the cells that produce red blood cells. 2. CRISPR technology also raises the possibility of manipulating species in the wild. CRISPR can be associated with a process called a gene drive. This is a system which allows a parent to pass on a characteristic to all offspring instead of just half. Currently, scientists are studying the possibility of using these CRISPR-based gene drives in mosquitoes. Genes which cause resistance to the malaria pathogen are inserted using the CRISPR-Cas9 system, and the gene drive ensures that an engineered mosquito would pass on its new resistance to all of its offspring. These mutant mosquitoes could potentially wipe out malaria for good in some regions of the world, a change that could save some of the 500,000 children who currently die of malaria each year. 3. Gene editing could also be used for biotechnology. It could be used in agriculture to genetically modify crops so that their yields are increased, they become resistant to disease, and they grow better in difficult climates. This could potentially improve food production across the world. Why hasn't it revolutionised our lives yet? Well, gene editing hasn't actually been around that long, and it wasn't until 2012 that the CRISPR system was demonstrated to work in gene editing. In 2013, CRISPR-Cas9 was shown to work in human cells, which triggered an explosion in research using this technology. But every breakthrough brings more and more ethical and practical arguments concerning the use of gene editing systems. Perhaps the most well-known cause for concern is the possibility of designer babies. Potentially, the genes of an embryo could be edited to change characteristics that have no bearing on health, for example eye colour. Arguments include the fact that it would be irresponsible to edit human genes which would be passed on to future generations, especially because not all relevant safety issues have been resolved. The ethical arguments include the fact that the idea of designer babies could open the door to the potentially scary practice of eugenics, where the human race is improved by controlling genetics and reproduction. Also, CRISPR can have some off-target effects, and although these effects are only a small percent, the changes we make to DNA could lead to more genetic complications, some of which could be fatal. Outside of human DNA, the possibility of creating more virulent microbial diseases could cause huge damage to the human lives by increasing their resistance to treatment.
To sum it all up, CRISPR has its advantages and disadvantages, ranging from ethical concerns to being known as the fastest, cheapest, and most precise way of editing genes. This scientific breakthrough has been taken up widely and spread quickly, as it's the ability to potentially eliminate diseases and solve world hunger, all by editing DNA. In the years to come, it certainly seems possible that our lives will be revolutionised by the advances being made in this currently small area of research.